Okay, let's go ahead and begin. Go ahead, Peter.
Every breath we take is modulated by nerves called respiratory chemoreceptors that detect chemistry changes in oxygen and carbon dioxide in our blood and send impulses to brain centers that determine how we breathe, how deeply and how often. Most of the time, normally our chemoreceptors detect any buildup of carbon dioxide, and this provides our main drive to breathe. Sadly, for some who require mechanical ventilation in a hospital, this drive to breathe that's automatic can be lost. This occurs most often with people who have advanced cardiopulmonary distress like heart failure or emphysema, but it can also occur in the ICU when someone has had an accident, or in some cases with premature infants who fail to wean from the ventilator. These individuals' chemoreceptors have reset to accept a high level of carbon dioxide in the blood as normal, and they're unable to sustain their own breathing for more than a few hours. While this threatens their life daily, there's no known treatment. My research focuses on a chemoreceptor found in the lungs of ducks and other birds called an IPC, or an intrapulmonary chemoreceptor. It's very similar to mammalian chemoreceptors, and it's easier to find and study. My research treats these chemoreceptors in a new way. Instead of looking at them as rigid structures which simply open and close ion channels, I've proposed that the chemoreceptor may change its volume in response to fluxes of ions into and out of the cell, and that specifically the negative ion chloride plays a key role in the nerve's ability to do this. Now we know that carbon dioxide passes freely through the cell membrane, and that it interacts with other ions to generate nerve impulses or action potentials. So by testing the effects of different drugs on ion channels, we are able to record the IPC nerve's results and responses to these treatments in real time, like you see here with frequency of action potential graphed against time. So the negative ion chloride has been studied very little in physiology, and my research has shown that by blocking certain chloride channels, the nerve either speeds up or slows down. And so this suggests that the nerve, because of the type of ion channel, may indeed be changing its volume, but more importantly perhaps, it also suggests that targeting chloride channels in people who are bordering on ventilator dependency in a hospital may be triggered to automatically breathe more often. We are exploring new territory with nerves and negative ions in the cell, and it's very exciting that we may be able to help people who could otherwise be dependent on a mechanical ventilator and supplemental oxygen for the rest of their lives. Thank you.